Thank you, Mr. Moderator, for a nice and a quick introduction. And as you mentioned, my name is Sintayo Ficado. And the title of my today's presentation is Infection-Associated Gastric Cancer, specifically the role of Helicobacter pylori and Epstein-Barr virus will be discussed. And this was my PhD work while I was in Japan. And I will present the findings and the research that I did while I was in Japan, actually. And finally, currently now I am extending my research related to H. pylori, Epstein-Barr virus, and so forth. So lately I will discuss how I am linking my previous knowledge and the research experience with the current research interests. So in order to go through my presentation, I have a list of contents. I will go through a brief introduction about myself, Hawassa University and Shimane University. Then I will talk about epidemiology of gastric cancer, and I briefly discuss about Helicobacter, what kind of bacteria is this, and what is Epstein-Barr virus, and what is Epstein virus associated gastric cancer. And then I will discuss about material and methods of the research, the main findings or results with discussions and conclusion, and finally with research areas for collaboration for the future. So in order to start with my introduction, as already mentioned, I've graduated in bachelor degree in 2005 in medical laboratory technology from Jimma University, Ethiopia. And I pursued my medical microbiology master's degree in 2011 from Addis Ababa University, and pursued further in medical microbiology and got PhD in 2021 from Shimane University, Japan. And almost 17 years since I have been in Hawassa University, in between study and working, in total of 17 years, 17. And I started working in Hawassa University as a graduate assistant level 2, and promoted like assistant lecturer, and currently I am serving as assistant professor in the Department of Microbiology and School of Medical Laboratory Technology. So Hawassa University, as all you know, Ethiopia is located in East Africa and the Horn of Africa, and one of the highly populated nation in Africa next to Nigeria, actually. And Hawassa University is located in the southern part of Ethiopia. And this university was established in 1976, and it's considered as the first generation university in Ethiopia, and organized in 9 colleges, including College of Health Science and Medicine, where I'm working now, and has 3 institutes as well. And the major activities of the university is teaching undergraduate and postgraduate students, including the master's and the PhD students. And recently, because of the scale up of the work of the university, it is differentiated as one of research university in Ethiopia. So regarding about Shimane University established in 1949, and this university is found in Japan, actually. And you know, Japan is one of the countries in Asia. And the university was a public university, actually, and engaged in undergraduate and postgraduate teachings, and one of the center for biomedical research in Shimane Prefecture or region. And I was attended PhD from 2017 to 2021. So the incidence of gastric cancer and gastrointestinal cancers this is worldwide problem and disease condition, and the incidence is so high which is like colorectal cancer is coming the first stop, and followed by stomach cancer in the form of incidence, and comparatively the mortality rate also followed by colorectal cancer, and so many individuals worldwide affected, and at the same time died because of these cancer conditions. So publicly, it is one of important research and researchable problem. So regarding gastric cancer, according to the Global Burden of Cancer 2020 report, it's mainly dominated in Asian countries, particularly in China. Otherwise, distribution is worldwide, but the reporting of this disease varies from the region to region, actually. And in some regions may not have enough data to report, and because of this, it seems like low prevalent, but in highly reporting countries like Asian countries, the disease condition is very prevalent and very high. And the main associated infections with gastric cancer is Helicobacter pylori and Epstein-Barr virus. And in the coming few slides, I will mention or describe about these pathogens. So Helicobacter pylori is a gram-negative bacteria, enteric bacteria, and it is one of group 1 carcinogen, meaning infection will lead to development of cancer, and WHO assigned in 1994 as one of carcinogenic bacteria. And the infection of H. pylori acquired in early childhood, particularly in low sanitary condition and close family contacts. And the bacteria has several virulent factors, some of the structural proteins or structural components, including flagella, adhesive proteins, and the type 4 secretion system. And some are secreted molecules like cytotoxin-associated gene A and vacuolating cytotoxin gene like VAC-A. These are secreted molecules which is injected into the epithelial cells and will have effect, oncogenic effect on the infected cells, actually. So everyone born with normal gastric mucosa, actually, and through time there is exposure to the bacteria and infection, actually, and this infection will result in acute gastritis, which is a response of infection, and this gastritis, acute gastritis, will be advanced form of chronic gastritis, and the chronic gastritis also develop into atrophic gastritis, intestinal metaplasia, dysplasia, and gastric cancer. Actually, this is a long process and described as a chorea cascade of H. pylori induced gastric cancer development, actually. And the second pathogen, which is recognized as formation for gastric cancer, is Epstein-Barr virus. As you know, these viruses are double-stranded DNA virus and under the family herpes viridae family, and almost greater than 90% of the world population infected with this virus. And the virus has two kind of life cycle. One is lytic replication, and the second is latent infection. And during latency, the viral genome, which is called circular and called episome, and this episome will be anchored to chromosomal the chromosomes of the host cells through EBNA1 viral proteins, actually. And EBV-associated gastric cancer assumes that originate from a single EBV infected cells through monoclonal proliferation. And this is a picture of patients with EBV-associated gastric cancer under endoscopy. So more molecular features of gastric cancer, EBV-associated gastric cancer, described recently by Cancer Genome Atlas Research Network in 2014. And they identified that this kind of cancer shows that PIK3CA mutation, actually this is uncontrolled proliferation of EBV infected cells, And again, it has overexpression of PDL1 or 2, and PDL1 antagonistics is the cancer cells for surveillance and immunological killing. The other is EBV CpG island methylterphenotype and heavily methylated host genome, so that expression of important host genes will be suppressed, and CDKN2A silencing. This is the cell cycle protein, check proteins, and uncontrolled cell cycle will progress, and even it is related to some immune cell signaling. So gastric cancer formation and H. pylori association with EBV was previously demonstrated by some researchers, and as you know, H. pylori related gastric cancer predominate in the anatome of gastric tissue, but EBV associated gastric cancer dominate in the fundus or body part of the gastric tissue. And the research they showed that a little bit older paper, they found that EBV-associated gastric cancer near to the border of atrophic gastritis with inflammatory response, and here is the concludes that during H. pylori infection, there is an inflammatory response, and inflammatory response will drive EBV carrying lymphocyte to site of inflammation. And that's EBV from the lymphocyte will be enter into epithelial cells and help for formation of gastric cancer actually. And recent report, they found that they took biopsy tissue from gastric cancer patients with different gastritis level like mild, moderate, and severe, and try to correlate with EBV copy number. And they found that EBV copy number is significantly associated with patients with moderate atrophic gastritis. So is there cooperativity between these two pathogens? This is a question for research, and there are two models regarding this hypothesis. The first model developed by Saju and his colleagues and published in Nature Microbiology in 2016, they said that EBV infects first epithelial cells, and viral oncogenic proteins or genes will heavily methylate the host genome. And this methylation will suppress expression of important genes like cellular phosphatase, SHP1, which is a protective one, and SH2P will be more, this is proto-oncogene actually, relatively high expression. And during H. pylori infection, KAGA protein of H. pylori will associate with SHP2 and drive carcinogenesis process. This is one of the model. The second model says that H. pylori infects gastric epithelial cell first and modify the gastric environment or gastric epithelial cells that will drive infection of Epstein-Barr virus and helps for development of EBV AGC. And in this study, we used the second model. So the research hypothesis and the objectives. Hypothesis is H. pylori enhances EBV infectivity, thereby contribute for the development of EBV AGC. And the objective is to assess the effect of H. pylori exposure to gastric epithelial cells for subsequent EBV infectivity. So regarding the materials and the methods, we use bacteria and cells in this research. These cells are gastric epithelial cells, actually cancer cell lines, AGS and MKN28. And we use also immortalized cell line GUS1, which is more physiologically mimic with normal gastric epithelial cells. And the cells was cultured in standard culture media RPMI 6040 with presence of 10% fetal bovine serum, and in the presence of antibiotics, penicillin, streptomycin, incubated at 37 degrees Celsius and 5% carbon dioxide condition. Regarding bacterial culture, I used different bacterial strains like CPY6271, and it is isogenic KAGE VAC mutant, CPY3401, and isogenic FLA mutant, TN2 and isogenic KAG-PI mutant. And bacteria was cultured in Brucella agar or brose in the presence of 5% heat inactivated horse serum and antibiotics like vancomycin, kanamycin, and chloramphenicol. So H. pylori is a microorophilic bacteria and requires a special condition for culturing under lab condition. So what we use is to make microphilic environment is we use an aeropack microaero, which is produced by Mitsubishi Gas Chemical Company in Tokyo, Japan. And these sachets will be inserted with a plate in the incubating jar, and immediately insertion, the oxygen will be utilized by the chemical system and reduced into around 8%, and similarly increasing the carbon dioxide concentration up to 5% actually. So when we culture in this condition, within 3 to 5 days, colonies will appear, and we will take the colonies and transfer into Brucella brose and culture under microorophilic condition for 18 hours. And after 18 hours, best or good, physiologically good bacteria will be measured, its OD, and the colony forming unit for infection will be adjusted. Next, cell-free EBV virus production. And in this research, we use Akata EGFP EBV recombinant virus actually. And the recombinant is we replace the thymidine kinase gene of the virus and insert neomycin resistance gene, which is used for selection purpose, and EGFP for reporting assay or reporting during infection assay. So latently infected AGS EBV will be transfected with BZLF1 transfection using lipofactamin 2000, and after 48 hours, we collect supernatant and filter sterilize, then concentrate at high-speed centrifugation, and finally suspend the virus in a cell culture medium and store at -18 until infection was performed. So establishing co-infection protocol, and we use gastric epithelial cells and the seed on a cell culture plate and allowed to attach for 12 hours. Then after 12 hours, we infect with H. pylori of MOI of infection 100 for 7 hours. Then after 7 hours, we remove bacteria and wash by phosphate buffered saline or PBS minus, and infect with cell-free EGFP EBV with MOI of 125 for 48 hours or 2 days. Then after 2 days, we collect the cell and examine with fluorescent microscope, the fluorescent activated cell sorting or FACS analysis, and the quantitative PCR or qPCR. So using the protocol, infection protocol, we examine the cells using microscope, and we found that the brightness of green fluorescent protein in H. pylori treated cells is higher than or more brighter than control one or EBV infection alone. So we examined this qualitatively and further analyzed by quantitative analysis using FACS and the qPCR, and we found that H. pylori treatment enhanced EBV infection efficiency in AJ cells. So in order to prove this phenomena is common for other gastric epithelial cells, we repeated the experiment in MKN28 cells and GUS1 cell. As I've said, GUS1 cell is immortalized cell and more mimic with normal gastric epithelial cells. And we found that similar data or result with AJ cells. So as a short summary, H. pylori pretreatment of gastric epithelial cells enhance EBV infection efficiency. Now the question is, which H. pylori factor or factors mediate EBV infection enhancement? So H. pylori has several virulent factors, and like flagella for motility and adherence, type 4 secretion system, and bacterial byproducts, which is injected through these type 4 secretion systems, like ADP hepatos or genomic DNA of the bacteria. which even activates NF-kappa B immune signaling and responsible for inflammatory response, and KAGE injection, which associated with SHP2, and this again helps driving for tumorigenesis. Again, VAC-A for vacuolation of the infected cells actually. So we examined the role of KAGE, VAC-A, KAG-PI, and flagella in our research. So the mutant bacteria was generated by inserting these genes, like KAGE was replaced by kanamycin resistance, VAC-A gene was replaced by chloramphenicol resistance, and KAG-PI also chloramphenicol resistance gene. And we treat gastric epithelial cells with wild-type bacteria, KAG knockout bacteria, and VAC-A knockout bacteria, and infect with EBV. And we assess infection efficiency. So in the first graph here, as you see the data, no infection enhancement after using the knockout bacteria in both cases. That means KAGE and VAC-A knockouts. So it has no role in enhancement of infection in this case. So the second data here, we use wild-type bacteria and KAG-PI knockout bacteria. And in this case, KAG-PI knockout bacteria do not enhance EBV infection efficiency or infectivity, and this is related. So EBV infection enhancement was KAG-PI dependent manner. So in order to make further investigation, really, is it KAG-PI or other factors also involved? We checked the flagella protein, and flagellar protein is a major component, and we use flagella A knockout and wild-type bacteria for treatment of gastric epithelial cells. And we found that flagella knockout bacteria could not enhance EBV infection from this data. And further, we checked attachment of the bacteria on gastric epithelial cells using GIMSA staining, and we found that the adhesion level of H. pylori in fly knockout H. pylori is lower compared to that of wild type. So does H. pylori require direct contact with epithelial cell for enhancement or not? In order to confirm this, we use transwell assay in order to separate direct contact between bacteria and the cells and to sequester the bacteria. And after 7 hours, we again infected with EBV, and we couldn't found any enhancement both in wild type and flagella knockout bacteria. So in order to further investigate, we forcefully attach the bacteria to epithelial cells using centrifugation technique. And after centrifugation, and infect the cells with the virus, EBV. And we found in this case, both wild type and fly knockout bacteria enhance EBV infection efficiency in a similar manner. And further, we also checked that the attachment level of bacteria in gastric epithelial cells after GIMSA staining, and we found the attachment or adhesion is very comparable. So as a short summary, the adhesion of H. pylori to gastric epithelial cells is important for enhancement of EBV infection. So now the question is, how H. pylori promotes EBV infection of gastric epithelial cells? So there are two hypotheses. One, once the virus enters the cell, there may be replication of the virus within infected cells and increase the total copy number of the viral genome within infected cells. The second one, H. pylori may stimulate expression of some viral receptors on gastric epithelial cells and enhance entry into infected cells actually. So we first check the first phenomena here, and we use BZLF knockout virus for infection after H. pylori treatment, and we found that BZLF knockout virus also enhance EBV infection after H. pylori treatment. And further, again, we treat the cells with H. pylori and infect with EBV, and we try to check the expression level of BZLF1, lytic gene, in the control and the treated cells. So we didn't find any significant difference the BZLF1 gene expression in H. pylori treated and untreated cells. So we jump to the second hypothesis that probably increased entry of EBV maybe happen in this case. So we treat the cells in a similar fashion with H. pylori and infect with EBV for 4 hours only. Then after 4 hours, we wash the cell and extract surface-bound viruses. And we check the EBV copy number between H. pylori treated and untreated cells. And we found that significantly higher EBV copy number in H. pylori treated cells. So this shows that H. pylori promotes EBV entry to gastric epithelial cells. Now the question is, does H. pylori infection upregulate EBV receptor expression on gastric epithelial cells? So previous research shows that low affinity EBV receptors is expressed on gastric epithelial cells. Actually, the integrin families, epinephrine type A receptor 2 or EphA2, and non-methyl myosin heavy chain 2 or NMHC2A, and a neuropilin family of proteins expressed actually. So we want to check whether expression of these proteins is upregulated or not. So we treat the gastric epithelial cells with H. pylori for 7 hours, and we extracted messenger RNA from infected and untreated cells and compare the expression level. As you see from this data, the integrin family of proteins didn't upregulated in H. pylori treated cells compared to that of untreated cells. And we checked expression again in EphA2 and NMHC2A, and only EphA2 and NMHC2A messenger RNA level expressed in higher compared to untreated cells, and neuropilin 1 also didn't expressed. So after messenger RNA expression upregulation, we check whether this is also manifested in protein level expressions. So we treated the cell with H. pylori in a similar fashion, and we extracted the cell and check after staining by primary and secondary antibodies for the proteins, and we check whether protein expression is increased or not. So here is bold line is H. pylori treated, and the light one is untreated cells. So there is a shift of the cells and indicating that expression of protein both in EphA2 and NMHC2A. And this data further strengthened using Western blots, and we treat cells with H. pylori, wild type and CAG-PI knockouts, and check the expression level. And as you see from the data, the wild type increase EphA2 expression in Western blot, and NMHC2A also expressed in higher level compared to the knockout bacteria. So finally, we want to check whether this phenomena is really working or not using siRNA treatments. So we treat with the cell with H. pylori first, and then followed by siRNA treatment for interference. And this interference abolishes the H. pylori mediated EBV infection enhancement after the siRNA treatment. And you see here siRNA control, there is significant difference between treated and untreated cells, but with EphA siRNA treatment and NHMT siRNA treatment, there is no enhancement of EBV infection after H. pylori infections. So as a conclusion, H. pylori adheres to gastric epithelial cells via type 4 secretion system and induces expression of EphA2 and NMHC2A. Then EBV is thought to infect cells through these receptors. So this is the whole story of the research. And now the research areas for collaboration As you know, gastric cancer is a disease with a multi-factor like virulence of bacteria or H. pylori, diets and lifestyle of the individual, and the population genetics is very different and diverse. So epidemiology varies globally actually, and there is little known in Ethiopia and in East Africa. So this is areas of research collaboration, anyone who is interested. And again, H. pylori drug resistance increasing globally, and WHO categorized it as a priority pathogen actually. And recently we performed a systematic review and meta-analysis in H. pylori eradication rate in Africa actually, and we found that 79% of eradication rate using first-line eradication therapy. So again, gut microbiome and H. pylori adaptability is not studied well. And this is also one of the research areas if someone is interested. And in general, gastrointestinal infection, including diarrheal disease, also interests to me. And finally, I would like to acknowledge Professor Hinori Yoshiyama. He is my supervisor and head of the microbiology department at Shimane University. I also want to thank Otsuka Toshimi Scholarship Foundation, Kanhara Foundation, Japan, who supported financially during my scholarship and stay in Japan. And I would like also to thank Hawasa University to make this collaboration effective and help me to study in Japan. And again, I want to acknowledge Alberta University, who make this presentation possible and sharing my research findings. And finally, my family and friends who encouraged during my research studies and overall support. And thank you very much for listening to me. And now you can ask me questions and you can forward the comments. And thank you very much.